So as we were saying, we want techniques that allow us to compare our order of magnitude estimates, the qualitative picture, the approximations with some precise numbers. And for molecular census, meaning numbers about the number of proteins, lipids, biomolecules, etc., we want methodologies that allow us to accurately quantify these. Now, one of the numbers that we talked at length about is the protein abundance in a cell in E. coli. And the most powerful technique for examining protein abundance was 2D gel electrophoresis followed by mass spectrometry. So we're going to go to that first. What is 2D gel electrophoresis? So hopefully many of you at least have come across SDS page, as it is called, sodium dodecyl sulfate polyacrylamide gel electrophoresis. This is a one-dimensional gel electrophoresis. So what is 2D over here? It's two dimensions. The first dimension is a pH gradient using the isoelectric point of an amino acid to separate it. After that is done, this is done with usually a slab of gel material. So you separate along one axis. You turn that axis around and layer that gel onto a denaturant gel, which is nothing but SDS page that we referred to earlier, and separate now along the second axis. The first one isolates according to the PI, the isoelectric point. The second dimension isolates according to molecular weight. This leads you to have this kind of a picture that looks complex at first sight. But either through mass spectrometry or through antibody staining or many other methods, you can identify the exact nature of these proteins. That is how these circles have been drawn, identifying DNA-K, RPOB, EF2, and HNS as examples of 4 proteins from E. coli. What about mass spectrometry? So, in terms of methodology for protein isolation, mass spectrometry involves taking a protein sample, proteolytically digesting it, typically it would be trypsin, performing a 1 or 2 dimensional chromatography to separate the fragments, and run a mass spectrum on it. Gas phase fragmentation then allows us to see these beautiful peaks that you see in the right part of this figure, which in turn signatures of what polypeptides are present. The methodology of using these spectral peaks is based on optimizing the enzyme digestion so that the typical size is a few amino acids long of each fragment. This is sometimes referred to as a fingerprint, this spectrum, because it is characteristic for a given protein. But we don't end here. If you now feed it into a computer, which is connected to a search engine of a DNA protein database, then the software, typically designed for these kind of problems, is capable of doing peak picking, meaning identifying the peaks, matching them with the database, and predicting a result that is your expected protein sequence. This is essentially nothing but identifying the proteins. You can reverse this method and use this for a protein of a fixed band to also do protein sequencing, different from Edmond degradation, for example. So this is how we get proteins and their identity. By quantifying the amount of protein isolated, you can quantify its amount and therefore its concentration also, and therefore its abundance in molar units. Electron microscopy, on the other hand, which is again invented out of a curiosity exercise for discovery, involves shining an electron beam, a focused electron beam on objects, and detecting the diffracted electron waves. These are reconstructed into an image. This image shows you electron dense and electron transparent regions, and these are then interpreted spatially. The advantage is that it is far, far better resolved in nanometer scale than light microscopy. And that brings us, of course, to quantitative microscopy. This is a very popular method, partly because you get not just identity of proteins, for example here, but also their location. Not just location, but even their dynamics. This is very powerful, as you imagine, because unlike in the case of 2D electrophoresis and electron microscopy, these are fixed snapshots there from a specific time point. Whereas this sort of microscopy can give you a whole time series or a spatio-temporal map of the molecule of interest. Typically, this has been engineered using a tool which was developed after the discovery of the green fluorescent protein. We have spoken very briefly about green GFP as a typical protein. And the idea is to tag an existing gene in the open reading frame. You remember we talked about open reading frames as the coding sequences, with another gene, resulting in a transcription and followed by translation of a protein that is a fusion protein, as it is called. In this case, the protein is tagged with GFP here at its C terminus or COOH terminus. By using both hexahistidine and MX2 fusion expressions, in this group study published in 2003, the group of the authors Hu and company managed to annotate, in other words, identify the localization and perhaps speculate about the function of 6,234 ORFs in terms of open reading frames. This was done, of course, by introducing this genetic tagging method on the chromosome, on the yeast chromosome. The organellar localization and classification of genes then becomes very easy because you just look and you can find out. So, for example, in this image here, the gene of interest is tagged with GFP, the organelle of interest is tagged in RFP, and the co-localization shows that the two are exactly co-localized. You can see more about yeast genome engineering and quantitative microscopy on this website of yeastgfp.yeastgenome.org. This kind of tagging, besides giving you a qualitative picture of where things are localized, can also give you a quantitative picture. So the idea is to, as earlier, make a PCR product with unique targeting sequences, the tag and the selectable marker on a sequence, such that the unique targeting sequences go to a specific region downstream, for example here, of the gene. By homologous recombination, They integrate with the genome. And because we have chosen the start and stop codons correctly, they can produce a protein which is tagged. To do this with about 6,000 strains, detect whether the specific tagged proteins are actually detected by antibody, measure with a GFP fluorimeter the fluorescence, and localize the localization of the molecule by microscopy. But we can also look at the number of proteins as a function of the molecules per se, meaning to say, we get an idea of the abundance. So the most common abundance is 2 to the power 12, which is 4,096, roughly 4,000 molecules per cell. The correlation, in fact, can also be done, has been done by Ghemmaghami et al. In 2003, where all proteins were quantified and their abundance correlated with the abundance of mRNA. So the mRNA molecules per cell seem largely to linearly correlate and saturate for higher values with respect to proteins. The idea of all proteins versus essential subset is that if all proteins are put, however, you see an initial flat line, no change with RNA value, and then an increase, suggesting that there are some proteins for whom the copy number of RNA molecules increase does not result in an increase in protein number. Could be either due to downregulation or lack of correlation. Whereas for a subset, the essential proteins, as they're sometimes called, without which the cells would not function, divide and grow, this is a linear relation, meaning as much RNA there is, those many proteins there are. The abundance, meaning to say number of proteins per cell, ranges between 50 to 10 to the power 6, 1 million proteins per cell in yeast. The method used for quantification was tap tagging. The transcript level was estimated using microarrays. These are also sometimes called DNA chips or expression arrays. So you see that the combination of microscopy and microarrays can give us abundances as well as correlations. Lastly, I want to touch on a method of microscopy that is referred to as single molecule imaging. As the word suggests, single molecule imaging is based on microscopy using light at a scale of a single macromolecule, one protein or less. And we discussed that an average protein's diameter is about 2 nanometers, so about 5 nanometers is the diameter of a protein. Just to give you an order comparison of the size scales involved, the order of magnitude scale on this in this image points to millimeter scale for animals, small insects, through hair length, through mammalian cells, mitochondria, viruses at 100 nanometers, proteins and small molecules. This dashed line here is referred to as Ab's diffraction limit and is about 0.2 micrometers or 200 nanometers. Ab's diffraction limit is a number that was arrived at through physical derivation by Ernst von Haber in 1873, and it states that the distance in xy between two molecules, two point sources of light, that can be successfully resolved is equal to lambda by 2 times Na, where lambda is the wavelength and Na is the numerical aperture of the microscopic setup. Na is also equal to n times sine theta, and theta is the aperture angle, n is the refractive index of the imaging medium. Rayleigh modified this and came up with a simplification, perhaps, that this resolving distance d is equal to 0.6 times lambda by Na. If you ever go to Jena, as it is written in English, I urge you to visit the grave of Ernst Haber, a founding father of modern biological microscopy and a very important figure in identifying the limit to light resolution. It's very fitting also that his gravestone, Kabristan, Kabar, has the equation that he invented on it. You would always want to have something beautiful like this. Yeah? Your memorial should have the equation you invented. So in terms of single molecule imaging, the point we were trying to make here is that the physics of light do not permit us to distinguish between two objects smaller than 0.2 microns apart, approximately. Which means that if two molecules, which are only 10 nanometers in size, are 10 nanometers apart, you will only see a big blob. You will not see two molecules. That's what is the meaning of diffraction limit. So if you want to go beyond the diffraction limit, you need to do something. These four methods, activated light localization microscopy, a PALM, stochastic optical reconstruction microscopy, a STORM, and stimulated emission depletion microscopy, STED, these three methods, are also collectively called nanoscopy because, unlike microscopy, they are able to look at things at nanometer scale and separate between them. So, single molecule imaging, as such, can be performed, for example, using photoactivated localization microscopy, PALM, for which we need not a normal fluorescent protein like green fluorescent protein, but a photoactivable fluorescent proteins. Photoactivable fluorescent proteins are those that, with a specific wavelength of light that is not the excitation wavelength, they are activated, otherwise they are in an inactive state. And as they stay in the active state, if you image them, they will give you signal. After the passage of some time, these molecules will revert to their dark state. This dark state means you cannot see them. You cannot see them means they don't show up in a microscopic image, which means they're not there. The molecule is there, but the light is not there. And by cyclically activating and inactivating, or activating and waiting for the spontaneous inactivation of molecules, you can sample a region and get around part of the diffraction limit. This was a very clever innovation by Eric Betzig and Jennifer Lippincott-Schwartz. Eric Betzig was given the Nobel Prize for chemistry, along with Stefan Hell. What you're looking at in the image here is a single bacterial cell with the photoactivated fluorescent proteins showing the number of molecules that could be identified, and with the number of molecules being counted based on the PALM estimate compared to electron microscopy. So in the next segment, we're going to talk about timescales.